Much of molecular biology research is built on the study of how cells work. Cells are often thought of as the building blocks of our bodies, but cells themselves are made of many detailed components and complex mechanisms, like a mini universe. Much of the study of cells focuses on what causes them to change from one type of cell to another. For instance, the cells in your eye versus the cells in your skin, or how healthy cells turn into cancerous ones. Each cell is like a factory that performs different functions. A huge part of what makes factories different are the machines they contain. Cellular machinery is called proteins, but wait, generally cells contain the same DNA, so how do cells turn into different types? Sections of DNA called genes each encode for a protein. The genes are transcribed into a copied form called RNA, then the RNA is used as the template to build proteins. This process is the foundation of molecular biology. Here are 5 experiments to study this process that show up very often in nearly every biotechnology research paper. 1. qPCR. Quantitative Polymerase Chain Reaction. Polymerase chain reaction, or PCR, is a method of making copies of DNA to increase the amount in your sample. Why bother with this? Because copying the DNA allows us to get enough of it to study, but also we can use PCR to measure how much of one type of DNA we have compared to another type. To do this, we have a fluorescent molecule that glows in the presence of our DNA. We combine that with all the things needed for PCR: nucleotides, or the molecules that make up the DNA, and an enzyme, which is like a molecular robot designed by nature. Here, the enzyme is called DNA polymerase, and it uses our original DNA as a template to make more and more of it out of the nucleotides. Once enough copies are made, the fluorescent chemical can be seen with a special instrument. So the more DNA we started out with initially, the sooner we can see the fluorescent glow. We measure this in terms of how many times the enzyme copied all the DNA, or number of cycles. By comparing the number of cycles needed to see fluorescence for one sequence of DNA to another, we can see how much more of one sequence we started out with initially. That's the theory, but in practice, usually we aren't interested always in DNA, but rather RNA that the cell is producing, because that is what is used to make proteins. To look at RNA quantitatively, there's an additional step to convert it back to DNA, called complementary DNA, using a different enzyme. Then, by looking at the complementary DNA with qPCR, we can calculate how much more of one RNA type is being made compared to another. As a note, usually the other RNA type we are comparing to is a housekeeping gene, or a protein we know the cell is always making. In this way, we can calculate how RNA types change in response to drug treatments or across different cell types by monitoring the levels of our RNA we are interested in compared to the housekeeping gene. 2. Microarrays. Microarrays are also a means of looking at the types and quantities of RNA cells are transcribing. However, unlike qPCR, which only examines 1 or 2 markers at a time, a microarray looks at hundreds or even thousands of RNA types within the same sample. So why bother with qPCR then? Although a microarray gives a very broad or global view of what RNA transcripts a cell is making, it isn't as accurate as qPCR. Imagine this, sort of like counting the number of people with specific names on the globe. A very specific analysis would be counting the number of people named Joe. A global analysis would be counting the number of people named Joe, Sue, Bob, all the way up to a list of hundreds or thousands of names. To collect all of this data, we might have different signs with all the names lined up, and let people within a crowd self-sort into the line with their name. This would collect a lot of broad data, but some people might not see their name or choose not to participate, and so the data might not be fully accurate. Much the same way, a microarray basically consists of a panel with points that certain sequences stick to. In this way, we can get an idea of the amounts of each sequence, but not in quite as accurate a way as qPCR. 3. Fluorescence reporter. The previous 2 methods focused on examining RNA, a step on the way to protein production. But RNA might not always make it to become a protein. It can be destroyed along the way. So it's important to also look directly at the proteins themselves. Proteins are very small, of course, so even with a microscope, we need a few tricks to be able to see them. One way to do this is to attach a fluorescent molecule onto the protein, called a tag, that glows under the microscope. In this way, we can see where in the cells proteins are located and infer what they might be doing. A fluorescent tag is basically another protein that has the ability to glow when light of a certain wavelength is shined on it. This property was originally discovered in jellyfish, green fluorescent protein, but there are other proteins with other colors as well. But how do we get the fluorescent protein attached to the protein we want to observe? We can create cells with a fluorescence reporter. This is done genetically by providing cells with the DNA sequence for the protein we are interested in, followed by the sequence for the fluorescent protein. In this way, we've tricked the cell so that it makes the protein we're interested in with the fluorescent tag attached. Now we can easily see if our protein of interest is being made, and if so, where in the cell it's located. But it's good to have more than one way of looking at proteins. 4. Western blotting. This is a technique to look at the proteins cells are making. In this technique, the cellular membrane is destroyed using chemicals. This is called lysing the cells. Next, the proteins are separated by putting them in a block of gel made of pores that allow proteins to separate according to size. The proteins are helped to migrate through the gel using an electric current. This process is called electrophoresis. Larger proteins take longer to move through the gel and thus stay towards the top. Smaller proteins move faster and end up nearer the bottom. The proteins sort this way and appear as bands. Next, the proteins are transferred from the gel to a special paper, again using the help of electric currents that attract the proteins. Specific proteins can be seen on the paper by using immunostaining, which we'll talk about next. 5. Immunostaining. Immunostaining has a number of different applications in molecular biology. The basic principle is this: antibodies are structures that specifically recognize and attach to certain proteins, much like a key fitting a lock. In the immune system, they function to recognize foreign proteins on viruses or bacteria, but antibodies can form that attach to any protein. This is done by injecting a purified protein into an animal such as a rabbit or goat called the host species. The host's immune system recognizes the protein as foreign and starts forming antibodies that bind to it. These antibodies can be isolated from the host animal's blood and are called primary antibodies, since they bind to the protein you're interested in. By mixing the primary antibody with the proteins bound to your paper in Western blotting, or by exposing cells that have been attached to a microscope slide, we have primary antibodies stuck to our protein of interest. To see where the proteins are, we can then mix in a secondary antibody that can only bind to the primary antibody. But the secondary antibody contains a fluorescent tag, in the case of immunofluorescence microscopy, or for Western blot or other similar applications, the secondary antibody might be attached to a molecule like HRP, which becomes visible after a chemical reaction. In this way, proteins can be visualized and even quantities measured by how much fluorescent or chemical signal they emit in the form of light. I hope you found this video helpful for understanding scientific research papers. If so, please subscribe and spread the word to help this channel get off the ground. And don't forget,